So in this video, I'm going to be talking about B cell development and maturation. So to begin with, I'm just going to talk a little bit about B cells. And B cells are also known as B lymphocytes. So if you hear one of those words, if you hear B lymphocytes, that means B cell. They are a type of white blood cell of the lymphocyte subtype. And they function in humoral immunity component of the adaptive immune system by secreting antibodies. So in the adaptive immune system, they secrete our antibodies. And in mammals, B cells mature in the bone marrow. And then in birds, B cells mature in the bursa of Fabricus, which is a lymphoid organ. And then that's enough about the background of B cells. And so moving on to development of B cells develop from hematopoietic stem cells, or HSC, and they originate from bone marrow. So HSC just stands for hematopoietic stem cell. And HSCs first differentiate into multipotent progenitor cells, that also stands for MPP, and then common lymphoid progenitor cells, CLP. And then from here, their development into B cells occurs in many different stages, and each stage is marked by various gene expression patterns and immunoglobulin H chain and L chain gene arrangements. So B cells undergo two types of selection while developing in the bone marrow to ensure proper development, and those are positive and negative selection. So I'm going to go by each of those. So first is the positive selection, and this process occurs through antigen-independent signaling involving both the pre-BCR and the BCR. So there is independent and then there's also dependent, but positive selection does it through antigen-independent. If these receptors do not bind to their ligand, B cells do not receive the proper signaling and cease to develop. So if the receptor is not able to bind to their ligand, there won't be any more development and everything will stop and nothing will happen. And then moving on to negative selection, this occurs through the binding of self-antigen with the BCR. If the BCR can bind strongly to the self-antigen, then the B cell undergoes one of four fates, and those fates are clonal deletion, receptor editing, anergy, or ignorance. And ignorance basically means that the B cell will ignore the signal and continue on with development. It just doesn't care about what just happened, it's just going to continue anyway. This negative selection process leads to a state of central tolerance, in which the B cells don't bind with self-antigens present in the bone marrow at the time. And to complete immature B cell migration from the bone marrow to the spleen, as well as passing through two transitional stages, you have to go through T1 and T2, and I'm going to tell you what those are. Throughout their migration to the spleen and after spleen entry, they are considered T1 B cells. And then when they're in the spleen, T1 B cells transition to T2 B cells. So they start out as T1 B cells, and then when they get in the spleen, they transition to the T2 B cells. And then T2 B cells differentiate into either follicular B cells, or that also can be FO B cells, or marginal zone B cells, or MZ B cells, depending on which signal they receive through the BCR and other receptors. So depending on what signal those B cells receive, it depends on which path they'll take. They'll either take the follicular B cell or the marginal zone B cell. Once differentiated, they are now considered mature B cells. So once they have been put in their different categories, they are the mature B cells that we know. So moving from development to B cell maturation, so the principal function of B cells are to make antibodies against antigens, and also perform the role of antigen-presenting cells, or APCs, and then eventually develop into memory B cells after activation by antigen interaction. So when we talked about the memory B cells in the other chapters, those were the cells that helps our body to remember what kind of pathogen or sickness is in our body. So it helps to get our body used to what kind of thing is overtaking it, And so B cells want to eventually develop into memory B cells. And then B cell development occurs through several different stages, each stage representing a change in the genome content of the antibody. So an antibody is composed of two identical light and two identical heavy chains, and then light is L and heavy is H. And the genes specifying them are found in the variable region, or the V region, and the constant region, or the C region. In the heavy chain V region, there are three segments, V, D, and J, and they recombine randomly in a process called Vdj Recombination to produce a unique variable domain in the immunoglobulin of each individual B cell. So the different regions, like the three segments, the V, D, and J, will combine to make Vdj. So that's all combined together. And then when the B cell fails any step of this maturation process, it will die by a process called apoptosis, which is basically programmed cell death. And then it can also die by clonal deletion as well. And then this is just a picture that I thought was pretty cool. It just kind of has like an outline of what's going on. And then right here you have a mature naive B cell, and then it goes in, and this is the B cell, and then over here has one way, and then you have another way going up here. And then this way it actually leads to beneficial mutation and affinity maturation, and then this way leads to apoptosis. So all of these lead to programmed cell death. And then when it's going through maturation, there's selection and class switching, and then the T cell, and then it is a CLL, and then it can become a memory cell, and then it goes to the plasma cell, and it shows you like the path of some of the cells, and it shows you both paths. So the beneficial path and then the death.